Next story. Targeting KRAS for overcoming chemoresistance in PDAC. In a groundbreaking study published in Nature Cancer, Huang et al. Have discovered a new approach to target KRAS-driven cancers, such as pancreatic ductal adenocarcinoma, PDAC, by promoting KRAS degradation through a previously unrecognized endogenous regulator, ADAM9, a disintegrin and metalloproteinase domain 9(4). This novel mechanism of KRAS degradation could potentially offer significant benefits over direct pan-RAS inhibitors that often suffer from off-target effects and may circumvent resistance to mutant-specific inhibitors. PDAC is one of the most lethal malignancies, primarily driven by oncogenic mutations in the KRAS gene. Recent advances have enabled the development of strategies to target KRAS, directly or indirectly, in PDAC and other KRAS-driven cancers. However, concerns regarding resistance against mutant-specific drugs remain, particularly in response to monotherapy. Huang et al. Demonstrate that ADAM9 indirectly promotes KRAS stability through transcriptional repression of the plasminogen activator inhibitor 1, PI1, which binds to KRAS and the autophagosomal adapter protein LC3 to induce lysosomal KRAS degradation. PI1 functions as a selective autophagy receptor and also binds to other RAS isoforms independently of their mutational status, indicating a potential means for universal RAS inhibition. 4. The authors conducted an in silico small molecule screen and identified a selective small molecule ADAM9 inhibitor that promotes KRAS degradation by increasing PI1 levels and induces anti-tumor activity in multiple in vitro and in vivo PDAC model systems. Notably, ADAM9 inhibition enhanced sensitivity to the first-line agent gemcitabine and mitigated gemcitabine-induced chemoresistance. 4. This discovery comes at a critical time, as current FDA-approved selective KRAS inhibitors, such as sotorasib and adagrasib, specifically target the KRAS G12C variant but are of limited relevance for patients with PDAC, as only approximately 1% of these cancers harbor the KRAS G12C mutation. For the mutant versions more commonly found in PDAC, KRAS G12D and KRAS G12V, several specific inhibitors are being assessed in clinical trials and others are still in preclinical development. The study by Huang et al. Suggests that targeting the process regulating KRAS protein stability through selective degradation, as demonstrated by their research, could offer critical benefits over direct pan-RAS inhibitors. This approach may also circumvent resistance to mutant-specific inhibitors, caused by reactivation of the RAS pathway through either the acquisition of additional mutations, intratumoral KRAS mutation heterogeneity, or the compensatory activation of wild-type KRAS alleles. The findings by Huang et al. Contribute to the expanding field of RAS targeting strategies, which include proteolysis-targeting chimeras, PROTACs, ubiquitination-independent degradation using the chemical knockdown with affinity and degradation dynamics, CANDDY, technology, and chimeric toxins targeting RAS. These diverse molecular attributes and cellular processes highlight the potential of indirect KRAS targeting in addressing unresolved issues, such as rapid resistance to allele-specific inhibitors, unfavorable bioavailability of current RAS degraders, and improved selectivity. Source: Laura Leonhardt and Matthias Hebrock. KRAS degradation averts PDAC chemoresistance. Nat Cancer 5, 375-377, 2024. Less than DOI greater than. Next story. Unleashing the potential of grand T cells through checkpoint blockade. In a groundbreaking study published in Nature Cancer, Hans R. Widland and Lydia Lynch reveal that grand T cells, a type of immune cell that has previously been overlooked, play a crucial role in the effectiveness of cancer immunotherapy. The researchers found that a specific subset of these cells, V delta 1+, is associated with better responses to immune checkpoint blockade, ICB, therapy in melanoma tumors with a lower neoantigen load. ICB therapy has been a game-changer for patients with certain types of advanced stage cancers, but predicting which patients will benefit most from these treatments remains a significant challenge. Conventional wisdom suggests that ICB therapy is beneficial in cancers with a high tumor mutational burden, such as melanoma, non-small cell lung cancer, and gastric carcinoma. However, Widland and Lynch's study challenges this notion by demonstrating that V-delta-1 plus grand T cells are associated with improved responses to ICB in tumors with a lower neoantigen burden. Grand T cells are a type of T cell that does not typically recognize antigens presented by major histocompatibility complex, MHC, molecules. Instead, they recognize stress ligands on tumor cells via their NKG2D receptors and other natural killer-like receptors. The study found that ligand recognition by NKG2D may be sufficient to upregulate PD-1 on V-delta-1 plus cells, and engagement of PD-1 by PDL-1 may hinder some of their anti-tumor functions. Intriguingly, ICB therapy can unleash their potent cytotoxicity, which would occur in the absence of MHC class I and neoantigens presented by MHC class I. Furthermore, the study found that PD-1 expression on V-delta-1 plus T cells could be a result of chronic stimulation of the grand TCR or activation of NKG2D. However, it remains unclear whether PD-1 is upregulated on V-delta-1 plus T cells after antigen recognition or through activation via the recognition of stress ligands by NKG2D. Therefore, further investigation is needed to determine whether V-delta-1 plus cells in the tumor become exhausted in the classic sense, with diminished cytolytic effector function. In conclusion, Widland and Lynch's study sheds new light on the role of grand T cells in responses to ICB therapy. The researchers propose that V-delta-1 plus cells are particularly important in tumors with low neoantigen burden and in mismatch repair deficient tumors expressing mutant beta-2 microglobulin, beta-2M, that have exceedingly low antigen presentation by MHC class I and evade conventional surveillance by CD8 plus T cells. These findings have significant implications for the development of future cancer immunotherapies. Source: Less Than Nature Greater Than Next Story. Unlocking the Power of Primal Fear, How It Shields Against Infection, Next Story. Primal fear protects against infection, a new ancestral antiviral pathway. In a groundbreaking discovery, scientists have identified a new ancestral antiviral pathway, called FEAR, FACT-ETS1 antiviral response, that provides an interferon-independent antiviral response in mammals. This pathway, which has been counteracted by a conserved poxvirus protein, represents a critical antiviral pathway that must be successfully targeted by several viruses. The study, published in Nature Microbiology, reveals that the poxvirus protein A51 can block antiviral responses against RNA viruses in moth cells. This finding suggests the existence of a broad-acting antiviral response conserved across vertebrates and invertebrates. The researchers further discovered that A51 specifically interacts with monosumo-Y-lated SBT16, SBT16SUMO, a modification that remains poorly understood in terms of its functional impact on FACT activity. The FACT complex, which is conserved across eukaryotes, plays important roles in fundamental processes ranging from DNA replication and repair to transcription. However, the study found that A51 tethers SBT16 SUMO to microtubules and prevents it from translocating to the nucleus, where it engages sites of active transcription and induces expression of the transcription factor ETS1. Genetic depletion of either FACT components or ETS1 rescued the replication defects of poxvirus lacking the A51R gene in mammalian cells. Interestingly, the antiviral activity of FACT was not reliant on intact IFN responses in the host cell. Moreover, the authors found that A51 proteins from diverse poxviruses retain the ability to antagonize SBT16 SUMO, highlighting the evolutionary importance of disarming the fear pathway. The discovery of the fear pathway has opened up several research avenues. For example, what activates FACT, and how are FACT and SBT16 SUMOylation regulated in response to infection? The authors show that early poxvirus gene expression is required for FACT activation, but the precise nature of the viral ligands or PAMPs involved remains to be uncovered. Moreover, whether there are potential upstream components of this pathway is unclear. Further studies will undoubtedly start to unravel the secrets behind this ancestral response pathway that continues to protect us against infection to this day. Source: Less Than Nature Greater Than Next Story. The impact of climate change on timekeeping worldwide, how climate change is affecting global timekeeping. In a world where time is money, it's hard to imagine that something as seemingly unrelated as climate change could have an impact on our clocks. However, A recent study has shown that the melting of polar ice caps due to global warming is actually affecting the Earth's rotation, and in turn, our timekeeping. Since the Earth's rotation is not perfectly consistent, scientists have developed atomic clocks to provide a stable and accurate timekeeping standard. However, the atomic clock time needs to be occasionally adjusted to match the Earth's rotation, and this is done through the addition or subtraction of leap seconds. A recent study by Dr. Simon A. M. Garrett of the University of Glasgow has found that the melting of polar ice caps due to climate change is causing a slight slowing of the Earth's rotation, which is delaying plans to add a negative leap second. This means that human-induced climate change may delay plans to add a negative leap second to ensure the two align. In addition to this fascinating connection between climate change and timekeeping, there are many other intriguing scientific discoveries and advancements that are highlighted in the latest episode of the Nature podcast. For example, researchers have discovered an inflammatory pathway in neurons that responds to DNA damage and has a key role in the persistence of memories. This finding has implications for the development of new treatments for neurodegenerative disorders. Another study highlights the potential of waste wood as a sustainable material for 3D printing, which could have significant implications for the construction industry and efforts to reduce waste. The podcast also explores the effect of wind turbines on property values and the plans for the upcoming Simons Observatory, which will map the afterglow of the Big Bang in unprecedented detail. In conclusion, while the connection between climate change and timekeeping may seem tenuous at first glance, it is just one of many fascinating scientific discoveries and advancements that are highlighted in the latest episode of the Nature podcast. By exploring the latest research and developments in fields ranging from neuroscience to renewable energy, the Nature podcast provides a valuable resource for anyone interested in staying up to date with the latest scientific discoveries and advancements. Source: Less Than Nature Greater Than Next Story. Unveiling the link between RIPK1, necroptosis, and premature aging. Scientists have discovered that inhibiting necroptosis, a type of programmed cell death, can significantly extend the lifespan of mice with progeria, a rare genetic disorder that causes premature aging. The study, published in Nature Cell Biology, found that blocking the activity of the RIPK1 kinase, a crucial regulator of TNF signaling and cell death, and particularly its kinase function, led to a dramatic extension in survival of mice with progeria. Progeria, also known as Hutchinson-Gilford progeria syndrome, is a rare genetic disorder that causes children to age rapidly, resulting in a decreased lifespan of only a few years. The condition is caused by a genetic mutation that leads to the accumulation of a toxic protein called prelamin A on the nuclear membrane, which disrupts the nuclear membrane and causes systemic inflammation. Previous studies have shown that blocking inflammation, genetically or with drugs, can ameliorate many aspects of the progeria phenotype and extend life in mouse models. However, the new study by Wang and Silka is the first to demonstrate that inhibiting necroptosis, and particularly the activity of the RIPK1 kinase, can significantly extend the lifespan of mice with progeria. The researchers found that ZMPSDE24- mice on a RIPK1 kinase dead, or, to a slightly lesser extent, necroptosis-deficient background, RIPIK3- and MLKL-, had a dramatically extended survival. Specifically, the median survival of ZMPSDE24- mice was extended from 17 weeks to 31 weeks when RIPK1 kinase activity was blocked. Interestingly, the study also found that genetic blocking of RIPK1 kinase inhibition appeared to provide a greater survival advantage than genetic inhibition of necroptosis alone, and that this correlated with an effect on induction of inflammatory cytokines. The discovery that defects in the processing of lamin A lead to systemic inflammation and progeria syndromes has led to several potential therapeutic targets. However, the new study suggests that genetically blocking RIPK1 kinase activity, but also necroptosis, is a particularly effective approach in delaying symptoms in one mouse model of progeria. The study also highlights the potential of small molecule inhibitors of RIPK1 kinase activity as a treatment for progeria. The current U.S. Food and Drug Administration, FDA, approved treatment for progeria is the FTI lonafarnib, which can extend the life of patients by about 2 to 4 years. However, this treatment is associated with side effects, including nausea and vomiting. In contrast, a multitude of companies are developing specific RIPK1 kinase inhibitors, some of which have already demonstrated an excellent safety profile in clinical trials. Overall, the study by Wang and Silke sheds new light on the role of necroptosis and RIPK1 kinase activity in progeria and highlights the potential of RIPK1 kinase inhibitors as a promising treatment for this devastating condition. Source: Less Than Nature Greater Than Next Story. Drytech. Stay sweat-free with moisture-wicking wearable device. In a groundbreaking development for the world of wearable technology, a team of researchers has unveiled a new breathable platform that could significantly improve the comfort and functionality of these devices. The study, published in Nature, details a three-dimensional liquid diode that could pave the way for permeable electronics that can be easily integrated with cutting-edge technology. The problem with most wearable electronics is their inability to manage moisture, which can lead to discomfort, loss of signal quality, and even cause the device to fall off. While breathable electronics do exist, they are still in their infancy, and there is a pressing need for a technology that can be easily integrated with advanced electronics. The new platform, developed by a team of scientists from the University of Helsinki, addresses this issue by creating a breathable patch that can be used in conjunction with a variety of wearable devices. This patch, made up of a three-dimensional liquid diode, is not only breathable but also highly permeable, allowing it to effectively wick moisture away from the skin and keep the wearer dry. The researchers believe that this platform has the potential to revolutionize the wearable technology industry by allowing for the creation of comfortable and multifunctional devices. The breathable patch can be easily integrated with a wide range of electronic components, making it a versatile solution for a variety of applications, from fitness trackers and smartwatches to medical devices and more. The team at the University of Helsinki is not alone in their pursuit of better breathable electronics. Researchers around the world are working to create more comfortable and functional wearable devices, and this new platform could be a significant step forward in achieving that goal. The development of this new platform is a major breakthrough for the wearable technology industry, and it is likely that we will see a proliferation of new and improved devices that take advantage of this technology in the coming years. With the rise of the Internet of Things, IoT, and the increasing popularity of wearable devices, this platform has the potential to significantly improve the user experience and make these devices more practical and useful in everyday life. In conclusion, the new breathable platform developed by the researchers from the University of Helsinki is a game-changer for the wearable technology industry. The three-dimensional liquid diode technology offers a highly permeable and breathable solution for managing moisture, making it an ideal candidate for integration with a wide range of electronic devices. This technology has the potential to revolutionize the way we think about wearable electronics and could lead to a new generation of comfortable and multifunctional devices that are better suited to the needs of users. Source: Less Than Nature Greater Than Next Story. Melting ice delays leap second issue temporarily. In a fascinating turn of events, it appears that the melting of polar ice caps, a consequence of human-induced climate change, has delayed the need for the implementation of a negative leap second. This discovery has provided a reprieve for metrologists, who are tasked with the challenge of removing a leap second from UTC for the first time in history. The addition of leap seconds has been a crucial aspect of timekeeping, ensuring that the international time standard, known as Coordinated Universal Time, UTC, remains in alignment with Earth's rotation. However, the increasing frequency of leap seconds has caused challenges for modern technologies, such as satellite navigation and computer infrastructure. In response, metrologists propose that UTC be kept in line with Earth's rotation, but with a tolerance for adjustment increased to a value larger than 1 second. This proposal was adopted by the General Conference on Weights and Measures, CGPM, in 2022, delaying the need for any adjustment for at least another century. Meanwhile, the melting of polar ice caps due to human-induced climate change has caused a deceleration in Earth's rotation, effectively postponing the need for a negative leap second until at least 2029. This delay provides valuable time for researchers to evaluate the probability and related risks of implementing a negative leap second before 2035. The discovery of this delay has been welcomed by metrologists, who are keen to avoid any unnecessary risks associated with the implementation of a negative leap second. By carefully monitoring Earth's rotation and calculating the associated uncertainties, researchers can anticipate any such change and ensure the smooth functioning of modern technologies. In conclusion, while the melting of polar ice caps due to human-induced climate change poses significant challenges for our planet, it has inadvertently provided a solution to the challenge of leap seconds in timekeeping. The additional time gained will allow researchers to carefully evaluate the risks associated with the implementation of a negative leap second and ensure the continued smooth functioning of modern technologies. Source: Less than nature, greater than. Next story. Electrons' role in advancing optical communications. Next story. A revolutionary approach to optical communications. Electrons flip a switch on light. In a groundbreaking study published in Nature, scientists have developed a method to control the polarization of light using electrically controlled magnetization, paving the way for stable and energy-efficient information transfer. This new technique has the potential to revolutionize the field of optical communications by enabling the seamless integration of electronics, spintronics, and photonics. Traditional optical communication relies on adjusting the intensity of laser light to transmit information, a process that is fast but has inherent physical limits. The new approach, however, uses circularly polarized light, which can encode information in the orientation of electron spins. This method has the potential to be much quicker than switching between intensity levels, as it relies on changing the polarization states. The study, led by Degradation et al., used a layer of quantum dots embedded in a semiconductor material. Electrons were injected into this material, and their spins determined the polarization of the emitted light. The researchers controlled the orientation of the spins through a phenomenon known as magnetization switching, which was induced by a material property called spin-orbit torque. The injector channel, made up of a stack of ultra-thin layers of magnesium oxide, cobalt iron boron, tantalum, and chromium, exerted a torque on the magnetization, switching its direction. By placing the injector channel as close as possible to the quantum dot layer, the researchers achieved a high degree of circular polarization, with the polarization level changing from 31% to 31% when the magnetization was switched. The new technique has several advantages over traditional methods. Firstly, it enables the creation of light with intermediate polarization, which is key to the applicability of the approach for multi-level modulation in polarization-based optical communications. Secondly, it does not require an external magnetic field, making it practical and promising for real-world applications. However, some challenges remain. The researchers used a single layer of quantum dots, but several layers would be better for maximizing the amplification of light intensity. In addition, the extent to which the light emitted is circularly polarized could be increased by incorporating different materials into the injector channel. Despite these challenges, the study presents an exciting glimpse of a way to achieve superior information technologies at little cost to the environment. With further development, this technique could lead to a new era of high-speed and energy-efficient optical communications. Source: Less than nature, greater than. Next story. Unveiling the Southern Ocean's mighty current, a fascinating journey through history. The Southern Ocean's surprising supercurrent. The Southern Ocean, one of the most remote and inhospitable places on Earth, is home to a powerful current that plays a crucial role in the global climate system. The Antarctic Circumpolar Current, ACC, is a massive ocean current that encircles the Antarctic continent, transporting more than 100 million cubic meters of water per second. This current connects the Pacific, Indian, and Atlantic Ocean basins, making it a critical component of the global ocean circulation and a key player in regulating the planet's climate. A recent study published in the prestigious journal Nature has shed new light on the history of the ACC. The study, led by Dr. Fabian Lamy and an international team of scientists, reconstructed the strength of the ACC over the past 5 million years and made a startling discovery. Despite significant changes in Earth's temperature over this time, the strength of the ACC has remained remarkably stable. The researchers used sediment cores from 5 distinct sites in the central South Pacific sector of the Southern Ocean to reconstruct the ACC's strength. These sediment cores provided a detailed record of the ACC's past behavior, allowing the researchers to track its changes over time. The study found that the ACC has undergone significant variability on timescales associated with changes in Earth's orbit. However, despite these variations, the ACC's strength has remained relatively constant over the past 5 million years. This is a remarkable finding, as it suggests that the ACC has been able to maintain its strength in the face of significant changes in Earth's temperature. The study also found that the ACC's strength is determined by both the strength of westerly winds across the Southern Ocean and large-scale gradients in the density of its waters. During glacial periods, the ACC weakens due to changes in both of these factors. The westerly winds shift northwards, and the waters in the northern parts of the Southern Ocean cool, making them denser. This causes the ACC to weaken, as the denser waters in the north prevent the flow of water from the south. However, the study also found that the ACC underwent a general increase in strength between 5 million and 3 million years ago, during a period of cooling on Earth. This trend runs counter to the expectation that the ACC is weak during cool glacial states and strong during warmer interglacial states. The researchers suggest that this unexpected behavior may be due to processes resulting from the climatic conditions that characterized the warm Pliocene, before the establishment of a larger ice sheet on Antarctica and increased sea ice extent at the end of the Pliocene. The study raises several important questions about the ACC's behavior and its impact on the global climate system. For example, how representative are the South Pacific records of the other sectors of the Southern Ocean? Can the relative contribution of changes in wind versus density gradients be more explicitly untangled by using robust reconstructions of the evolution of north-south density gradients across sectors of the Southern Ocean? Finally, to what extent has the depth dependence of ocean current speeds in the ACC changed over time? These are important questions that need to be addressed in future research. However, the study provides valuable insights into the behavior of the ACC and its role in the global climate system. It highlights the importance of understanding the complex interactions between the oceans, the atmosphere, and the cryosphere in shaping our planet's climate. In conclusion, the Southern Ocean's ACC is a powerful current that plays a critical role in the global climate system. The recent study published in Nature has provided important insights into the ACC's history, revealing that despite significant changes in Earth's temperature, the ACC has remained surprisingly stable over the past 5 million years. However, the study also raises important questions about the ACC's behavior and its impact on the global climate system. As Earth's climate continues to change, it is crucial that we deepen our understanding of the ACC and its role in the global climate system. Source: Less Than Nature Greater Than Next Story. Sweat-proof wearable patch, your ultimate electronic companion. Imagine a world where your workout clothes can monitor your biometrics and the environment in real time, giving you instant feedback on your health and the conditions around you. This is no longer a distant dream, thanks to the development of a new wearable electronic patch that is impervious to sweat. In a recent study published in Nature, a team of researchers led by Zhang et al. Have created a flexible device that can rapidly discharge sweat, ensuring comfort and stability during prolonged wear. Current wearable electronic patches face significant challenges when it comes to managing sweat. While some designs use mesh-like electrodes without backing layers for breathability, this makes it difficult to attach the devices to the skin and integrate them with advanced electronics. Other designs incorporate permeable substrates with micrometer or nanometer-sized pores, but these openings can allow sweat to flow both ways, causing short circuits and corrosion. To solve these problems, Zhang et al. Engineered a wearable electronic patch by combining two liquid diodes, devices that allow liquid to flow in one direction only. The first component, a vertical liquid diode, is made of fabric that is hydrophilic on the top and hydrophobic on the bottom. This gradient in wettability facilitates unidirectional liquid transport, allowing sweat to move from the bottom to the top. A horizontal liquid diode on top of the vertical one uses superhydrophilic micropillars to drive sweat towards the edges of the patch, where it is released from outlets. A ring of polyester fabric between the two layers acts as a sweat collector, ensuring that any electronics integrated into the center of the substrate remain unaffected by perspiration. The researchers demonstrated the versatility of the patch by integrating it with various wearable electronic systems that were connected using detachable magnets. For example, when electrocardiogram (ECG) electronics were incorporated into the patch, it adhered to skin better and showed lower levels of signal interference than did existing ECG patches, providing stable heart rate readings over the course of a week. The authors also showed that they could integrate an electromyography (EMG) device into the patch, allowing for the measurement of electrical activity in muscles. The potential applications for this technology are vast. For instance, The patch could be incorporated into a T-shirt embedded with electronics that record real-time meteorological parameters, making the garment a wearable weather station. This could benefit outdoor enthusiasts and people working in challenging environments, even in heavily humid or rainy conditions. The patch could also be used in medical settings to monitor patients' biometrics or to deliver drugs through the skin. While further research is needed to make the current design suitable for extreme conditions, the potential for this technology is immense. The ability to create high-performance wearable patches or smart textiles that contain complex electronic components is an exciting prospect, bringing us one step closer to a world where our clothes are not just fashionable but also intelligent. Source: Less Than Nature Greater Than Next Story. Bee populations at risk. Climate change threatens further decline. Climate change threatens bee populations. A new study reveals the devastating impact on bumblebees and other bee species. Climate change is causing declines in bee populations, and a new study reveals the dire consequences for bumblebees and other bee species. The study, published in Nature, provides evidence of the direct physiological effects of extreme climate conditions on the long-term population stability of bees. According to the study, bumblebees are experiencing contractions in their dispersal ranges, population declines, and local extinctions due to a lack of physiological tolerance of high temperatures. Bumblebees are not the only bee species affected by climate change. A separate study published in the same issue of Nature found that most bee species, which are ecologically and evolutionarily different from bumblebees, are also experiencing declines due to climate change. These species are generally small and solitary, and they might respond differently to climate change than bumblebees do. The study, led by researchers at the University of California, Berkeley, used a 16-year study of 339 bee species, many of which are solitary, in drylands in the southwestern United States. The researchers used a robust combination of data for natural variation in climate and experimental evidence to predict which species would be negatively affected by climate change. They found that of the 243 species that were sensitive to drought, 46% will experience population declines with continuing climate change. The study also found that species predicted to persist despite climate change are tolerant not only of heat but also of dry conditions, desiccation. The authors experimentally determined thermal and desiccation tolerances for a subset of 12 bee species and found that those species that are best able to handle both were more resilient to previous climate change. The study's lead author, Dr. Claire Kramer, said, "Our findings suggest that species with a higher tolerance to heat and desiccation are better able to cope with climate change and that this trait is crucial for their survival." The two studies highlight the urgent need for conservation efforts to protect bee populations from the devastating impact of climate change. According to Dr. Kramer, bee populations are critical for the pollination of crops and wildflowers, and their decline could have serious consequences for food security and biodiversity. Conservation efforts must include the protection of bee habitats and the creation of climate refugia, which are areas where bees can find shelter from extreme climate conditions. Governments and conservation organizations must also develop strategies to help bee populations adapt to climate change. The study's findings emphasize the critical role that bees play in our ecosystem and the urgent need to protect them from the devastating impact of climate change. The loss of bee populations could have serious consequences for food security and biodiversity, and it is our responsibility to take action to protect them. References. Source: Less Than Nature Greater Than Next Story. Unveiling the Secrets of Lysosomal Organelles Through Fission, a competitive pursuit, in a groundbreaking discovery that could shed light on the fundamental processes that govern the functioning of cells. A team of researchers has identified the factors that enable the division of lysosomal organelles. The study, published in Nature, has revealed the crucial role of the protein MROH1 in facilitating the fission of lysosomes. Lysosomes are acidic organelles that play a critical role in the degradation and recycling of cellular waste. They also function as signaling hubs that regulate cellular functions and quality control. Maintaining the size and shape of lysosomes is essential for their proper functioning, and this is achieved through a delicate balance of fusion and fission events. While the process of fusion has been well studied, the mechanisms underlying fission have remained elusive until now. The researchers, led by Li et al., set out to identify the factors that mediate lysosomal fission. They began by examining the worm Caenorhabditis elegans and discovered that mutations in the gene HPO27 caused lysosomes to become extensively tubular. This coincided with a decrease in acidity and degradative capacity of the organelle, indicating that the shape of a lysosome is intricately linked to its functions. The mammalian protein MROH1 is equivalent to the protein encoded by the worm gene HPO27, and lysosomes look extensively tubular when MROH1 is removed from mammalian cells. The researchers found that the protein MROH1 is recruited to the lysosome through interactions with the protein RAB7 and specific lipids present on lysosomes. MROH1 assembles around tubules that emerge from the lysosome, mediating the fission of these tubules. An in vitro system consisting of artificial membrane nanotubes further confirmed the role of MROH1 in fission. The discovery of MROH1 as a lysosome fission factor has significant implications for our understanding of cellular processes. The finding that such a factor is both necessary and sufficient for lysosome fission provides an avenue for addressing some of the many questions that remain unanswered. This is because scientists now have a defined target that can be used to help establish links between lysosome functions and cellular and organismal physiology. The discovery of MROH1 as a lysosome fission factor is a notable observation that raises some exciting questions. The finding that such a factor is both necessary and sufficient for lysosome fission provides an avenue for addressing some of these questions. This is because scientists now have a defined target that can be used to help establish links between lysosome functions and cellular and organismal physiology. In summary, the study by Li et al. Has identified the crucial role of MROH1 in facilitating the fission of lysosomal organelles. This discovery is a significant step forward in our understanding of the fundamental processes that govern the functioning of cells, and it has the potential to open up new avenues of research in the field of cell biology. Source: Less Than Nature Greater Than Next Story. Maximizing social media impact on research, tweeting elevates engagement, falls short on citations, next story. The Great Twitter Experiment, debunking the myth of social media citations. In a world where social media dominates as a primary source of information, it's no surprise that scientists have turned to platforms like Twitter to share their research. However, a groundbreaking study published in PLOS One has revealed that tweeting about a research paper does not, in fact, lead to an increase in citations. The international team of researchers, led by marine ecologist Trevor Branch from the University of Washington, sought to uncover the truth behind the correlation between posting on social media and citation rates. They conducted an experiment spanning 10 months, where each researcher was allocated a randomly selected primary research article or review from a journal of their choice to post about on their personal Twitter account. The results were nothing short of surprising. Despite the increase in daily downloads for the posted papers and higher accumulated altmetric scores, there was no significant difference in citation rates between the posted articles and the control articles. These findings echo those of information scientist Stephanie Haustein at the University of Ottawa, whose 2013 study found a low correlation between posts and citations. Haustein notes that a significant issue with using posts as a metric is the presence of bots and noise in the signal. Moreover, with the recent departure of scientists from Twitter, the idea that posting could increase citations has taken a significant blow. The findings of this study are crucial for scientists and researchers who have long believed that tweeting about their work would lead to increased recognition and citations. Although social media remains a powerful tool for disseminating scientific information, it is clear that its impact on citations is minimal. The Great Twitter Experiment has provided valuable insights into the relationship between social media and academic research. While Twitter can help increase the visibility of research papers, it does not necessarily translate into higher citation rates. As the scientific community continues to grapple with the challenges of social media, it is essential to approach the use of these platforms with a critical eye. Sharing research on social media may have its benefits, but it is not the be-all and end-all of academic success. Source: Less Than Nature Greater Than Next Story. Climate Change Impacting Earth's Rotation. How Timekeeping Could Be Affected. Climate change slows Earth's rotation and delays leap second. Climate change is not only causing sea levels to rise and weather patterns to change but is also affecting the Earth's rotation, leading to a delay in the next leap second. According to a new study published in Nature, the melting of polar ice caps due to global warming has caused a shift in mass towards the equator, slowing down the Earth's rotation. The study predicts that the next leap second, initially scheduled for 2026, will now be pushed back to 2029. Duncan Agnew, a geophysicist at the Scripps Institution of Oceanography and author of the study, used mathematical models to analyze the contributions of geophysical phenomena to Earth's rotation and predict their effects on future leap seconds. Agnew's findings show that climate change has caused the Earth's rotation to slow down, requiring fewer leap seconds. Leap seconds are used to synchronize official time from atomic clocks with that based on Earth's unstable speed of rotation. The last leap second was added in 2016, and scientists had planned to add another one in 2026. However, the new study suggests that the next leap second will not be needed until 2029, thanks to the slowing down of the Earth's rotation caused by climate change. The slowing down of the Earth's rotation has significant implications for timekeeping in metrology. Leap seconds can cause havoc for computing systems, and a negative leap second, where a second is skipped, is expected to happen for the first time. This has caused concern among time metrologists, who are unsure of how to cope with a missing second. The slowing down of the Earth's rotation is caused by several geophysical phenomena, including core currents and the melting of polar ice caps. The melting of polar ice caps has caused the Earth to become less spherical and more flattened, leading to a shift in mass towards the equator. This shift has slowed down the Earth's rotation, leading to a delay in the next leap second. The study highlights the profound impact of human activities on climate change and the planet's rotation. Junli Chen, a geophysicist at the Hong Kong Polytechnic University, states that the postponing of a leap second is just one more example of how human activities have a significant impact on the planet. The delay in the leap second is good news for metrologists, who view it as a positive development. However, it is essential to note that the slowing down of the Earth's rotation is just one of the many negative consequences of climate change. The impact of climate change on the planet's rotation is a stark reminder of the urgent need to address global warming and reduce greenhouse gas emissions. In conclusion, the study highlights the profound impact of climate change on the Earth's rotation and timekeeping. The delay in the next leap second is a significant development, but it is crucial to remember that it is just one of the many consequences of global warming. It is essential to take urgent action to address climate change and reduce greenhouse gas emissions to prevent further negative consequences for the planet. Source: Less Than Nature Greater Than Next Story. Revitalizing the Immune System with Anti-Aging Antibodies. In a groundbreaking study published in Nature, researchers have discovered a new way to rejuvenate the immune system of aged mice using antibody therapy. The study found that depleting an expanding pool of aberrant stem cells in aged mice using antibody therapy can rebalance blood cell production, diminish age-associated inflammation and strengthen acquired immune responses. As we age, our immune system becomes less efficient, making us more susceptible to infections and chronic diseases. One of the reasons for this decline is the aging of hematopoietic stem cells, HSCs, which are responsible for producing blood cells throughout our lives. With age, there is a substantial increase in the proportion of HSCs that are biased towards generating myeloid lineage cells, which include red blood cells, platelets, and cells of the innate immune system. This imbalance in blood cell production results in poor adaptive immune responses to infection and high levels of inflammation, making us more vulnerable to diseases. To address this issue, the researchers took advantage of a key feature of myeloid-biased HSCs, a set of proteins expressed on their cell surface that makes them distinct from balanced HSCs. By mining transcriptional datasets, the authors identified 3 cell surface proteins that could be used to identify, isolate, and target myeloid-biased HSCs in aged mice, and potentially in humans, with minimal off-target effects. The researchers established an antibody conditioning protocol in which aged mice were treated with antibodies that targeted the cell surface proteins that the authors had identified. They found that antibody conditioning preferentially depleted myeloid-biased HSCs, and that balanced HSCs were mostly spared. Furthermore, they found that a single course of antibody conditioning was durable, with the relative number of myeloid-biased HSCs remaining depressed for at least 2 months after treatment. The study has significant implications for the development of new treatments for aging-related diseases. By rebalancing blood cell production and restoring adaptive immunity, depleting myeloid-biased HSCs in aged mice improved immunity in older adults, which could reduce infection-related morbidity and mortality. Moreover, the study raises the possibility that targeting myeloid-biased HSCs could alleviate some of the detrimental effects of aging by attenuating inflammaging and reducing the incidence and severity of diverse age-related inflammatory diseases. While the study offers a promising approach to rejuvenating the immune system of aged individuals, more research is needed to determine the safety and efficacy of this therapy in humans. However, the study provides hope for a future where we can delay the onset of aging-related diseases and improve the quality of life of older adults. Source: Less Than Nature Greater Than. Next story. How neurons' innate immunity strengthens memory retention. Next story. A surprising link between inflammation and memory. Innate immunity in neurons makes memories persist. Memory, a fundamental aspect of human consciousness, has been a subject of fascination for centuries. From surrealist paintings to cutting-edge neuroscience, the mystery of how memories persist has captivated minds and inspired art. Now, a groundbreaking study published in Nature has uncovered a population of neurons that engages mechanisms of the innate immune system during memory formation, revealing that inflammatory signaling might pave the way for long-term memory. The hippocampus, a seahorse-shaped region of the brain associated with memory and learning, has long been a focus for researchers. Decades ago, a patient known as HM underwent surgery to treat epilepsy, resulting in the removal of parts of his temporal lobes, including the hippocampus. While the surgery was successful in controlling his seizures, HM was left with a devastating side effect: he could no longer form new memories. This case led scientists to hone in on the hippocampus as a hub for many types of memory, sparking a flurry of research into the mechanisms that underlie memory formation and persistence. In the new study, scientists investigated neurons in the hippocampus of mice following memory acquisition. They discovered a distinct population of neurons that showed an inflammatory response to DNA damage, persisting for days after memory acquisition. This inflammatory response was mediated by a protein of the innate immune system called TLR9, Toll-like receptor 9, which detects extranuclear fragments of DNA that have been incorporated into cellular compartments called endolysosomes. The researchers demonstrated that disrupting these mechanisms, specifically removing TLR9 from hippocampal neurons, prevented mice from recalling long-term memories. This suggests that the sensing of double-strand DNA breaks by TLR9 is a versatile molecular process that is active in both memory and innate immunity. The authors of the study also showed that removing a protein called RELA, which connects TLR9 activation to DNA repair by 5,3-BP1, interferes with the accumulation of the centrosomal DNA damage response complex. This is a key finding, as the accumulation of centrosomal DNA damage response complexes is crucial for maintaining genome integrity. These findings have significant implications for our understanding of memory formation and the role of the innate immune system in the brain. They also provide a molecular link between the acquisition of a memory and the ability to store it reliably for an extended period. Despite these substantial insights, pertinent questions remain. For example, what properties of memory are conferred by these neurons, and how do they interact with engram neurons, which are known to be required for memory formation? The authors speculate that inflammatory neurons could have a key role in the stability and flexibility of memories, but that engram neurons could enable the recall of memories. Perhaps the engram neurons produce the initial memory signal, and the inflammatory neurons identified in this work support and sculpt the memory, enabling its persistence. However, the activity or functional properties of these neurons during the acquisition, consolidation, or retrieval of memories remain undefined. Recordings of electrical activity and selective manipulation of inflammatory neurons throughout the memory process will be needed to better understand the cell's contributions to memory persistence. This study raises pressing questions regarding the relationship between DNA damage, inflammation, and memory. Activation of TLR9, a key player in the immune response, is known to contribute to neurodegeneration, predominantly through microglia, the immune cells of the central nervous system. How is it that, in neurons, activation of TLR9 is crucial for memory formation, whereas, in microglia, it produces neurodegeneration, the antithesis of memory? What separates detrimental DNA damage and inflammation from that which is essential for memory? And how are these mechanisms disrupted in the context of aging and neurodegenerative disorders? The discovery of the role of inflammatory neurons in memory formation provides a fascinating new avenue for understanding the complexity of the brain and memory. As researchers continue to unravel the intricate network of interactions between neurons and the immune system, we can look forward to a deeper understanding of how memories are formed, maintained, and retrieved. Source: Joe Vasovich, V. al. 2024. Formation of memory assemblies through the DNA sensing TLR9 pathway. Nature. Less than DOI greater than next story. The controversial Harvard study on sun dimming. What comes next? The sun dimming experiment, known as the Stratospheric Controlled Perturbation Experiment, SCOPEX, was set to be the first to test the injection of particles into the Earth's upper atmosphere to measure their ability to reflect sunlight back into space. The experiment aimed to determine if such a technique could provide emergency relief from the worst impacts of climate change, as governments struggle to limit greenhouse gas emissions. However, the study faced opposition from those concerned about potential unintended consequences and the lack of international consensus on solar geoengineering. Opponents also worry that it could reduce pressure on world leaders to eliminate greenhouse gas emissions by offering a Plan B. The Harvard team had planned to launch a high-altitude balloon into the stratosphere, releasing up to 2 kilograms of calcium carbonate particles, and then measure their dispersal, interaction with other chemicals, and ability to reflect sunlight. However, the first launch, intended as an equipment test and set to take place at the Esrange Space Center in northern Sweden, was called off in 2021 after environmentalists and local indigenous groups announced their opposition. The cancellation of the Harvard study, however, has not halted all research in this field. Independent researcher Andrew Lockley launched a low-cost balloon that released 400 grams of sulfur dioxide into the stratosphere in 2022, and a for-profit company called Make Sunsets has begun dispersing sulfur dioxide into the stratosphere by balloon. Meanwhile, the U.S. National Oceanic and Atmospheric Administration, NOAA, has begun gathering background data from the stratosphere to better understand potential solar geoengineering efforts. Despite the controversy, scientists say that discussions about solar geoengineering aren't going away. The World Climate Research Program has launched an initiative to promote research into climate interventions such as solar geoengineering, and scientists are working to build the world's largest academic initiative focused on climate engineering at the University of Chicago. While some argue that such experiments should not proceed without broad public input, others argue that small experiments that are not expected to impact the environment should not require a global process. As climate change continues to pose significant threats to the planet, the debate over sun dimming experiments and solar geoengineering is likely to continue. Source: Less than nature greater than next story. Revitalize your immune system, strategies to restore youthful vigor. In a groundbreaking study, researchers have found a way to rejuvenate the immune systems of older mice, making them more comparable to those of younger mice. This was achieved by reducing the number of aberrant stem cells that generate a variety of other cell types, including those that contribute to inflammation. The research, published in Nature, has the potential to significantly impact the elderly population, who are more prone to severe infections and have weaker responses to vaccination. The study, conducted by researchers from Stanford University, discovered that as mice age, the balance between two types of blood stem cells shifts. In younger mice, there is a balance between the two types, but in older mice, the balance becomes skewed towards the pro-inflammatory innate immune cells. This shift leads to a diminished ability to mount new antibody and T-cell responses. To test if restoring balance to the populations of blood stem cells could rejuvenate the immune system, the team generated antibodies that bind to the blood stem cells that predominantly generate innate immune cells. They then infused these antibodies into older mice, hoping that the immune system would destroy the stem cells bound by the antibodies. The antibody treatment was successful and rejuvenated the immune systems of the treated mice. The treated mice had a stronger reaction to vaccination and were better able to fend off viral infection than older mice who had not received the treatment. The treated mice also produced lower levels of proteins associated with inflammation than did old, untreated mice. While the research is still in its early stages and has not yet been tested in humans, the study's findings offer a promising new approach to treating age-related immune system decline. The research team is planning to continue studying mice to learn more about other effects of the antibody therapy, such as whether it affects the rates of cancer or inflammatory diseases. The study's findings offer hope for the elderly population, who are more prone to severe infections and have weaker responses to vaccination. With further research, this new approach could have significant implications for human health, particularly for those in the older age groups. Source: Less Than Nature Greater Than Next Story. Unlocking Memories, the Intriguing Science of DNA Repair. In a groundbreaking study, scientists have discovered that the formation of long-term memories is linked to the breaking and repairing of DNA in nerve cells, a process that is facilitated by an inflammatory response. The researchers found that when a long-term memory is formed, certain brain cells experience a surge of electrical activity that causes DNA damage. This damage is then repaired by an inflammatory response, which helps to cement the memory in place. The findings, published in the journal Nature, suggest that this DNA damage and repair cycle plays a crucial role in the formation and durability of memories. These results provide the best evidence so far that DNA repair is important for memory, said Thomas Ryan, an engram neuroscientist at Trinity College Dublin. But it's still unclear whether the DNA damage is a consequence of engram creation, or if it encodes something distinct from the engram. The researchers trained mice to associate a small electric shock with a new environment, and then examined gene activity in neurons in the hippocampus, a brain area crucial for memory. They found that some genes responsible for inflammation were active in a set of neurons 4 days after training, but were much less active 3 weeks later. Further investigation revealed that the cause of the inflammation was a protein called TLR9, which triggers an immune response to DNA fragments floating inside cells. In this case, the nerve cells were responding to their own DNA, rather than to invading pathogens. The researchers found that TLR9 was most active in a subset of hippocampal neurons where DNA breaks resisted repair. These neurons also had accumulations of DNA repair machinery in the centrosome, an organelle that is typically associated with cell division and differentiation. However, mature neurons do not divide, so it is unclear why the centrosome is participating in DNA repair. When the researchers deleted the gene encoding the TLR9 protein from mice, the animals had trouble recalling long-term memories about their training. These findings suggest that we are using our own DNA as a signaling system to retain information over a long time, said study co-author Jelena Radulovic. The study also raises the possibility that this DNA damage and repair cycle might be faulty in people with neurodegenerative diseases such as Alzheimer's, leading to a buildup of errors in a neuron's DNA. These results bring much-needed attention to mechanisms of memory formation and persistence inside cells, said Clara Ortega de San Luis, a neuroscientist who works with Ryan at Trinity College Dublin. We know a lot about connectivity between neurons and neural plasticity, but not nearly as much about what happens inside neurons. Source: Less Than Nature Greater Than Next Story. The impact of mass resignations by journal editors. A new wave of collective resignations is sweeping through the academic world, as journal editors are increasingly stepping down from their posts en masse. The latest incident occurred earlier this month when the editors of Syntax, a linguistics journal published by Wiley, resigned in response to changes in the manuscript handling process that they felt would compromise the quality of the journal. According to a tally by the website Retraction Watch, the Syntax editors' resignations were the 5th such event this year and the 12th since 2021. The wave of resignations appears to be a response to changes in the business models of academic publishers, with many editors objecting to the article processing charge model, in which authors are charged fees to publish their papers open access. The resignations are not just a form of protest, but also an attempt by editors to take control of the scholarly communication process. In some cases, groups of editors who resign go on to found new publications over which they have more control. For example, the former editors of Critical Public Health, who resigned last year in protest against the publisher's plans to make the journal open access, are in the process of setting up a new journal called the Journal of Critical Public Health. Similarly, the former editors of Elsevier journal Neuroimage, who resigned last year, have set up another journal hosted by the non-profit publisher MIT Press. Stephen Smith, a biomedical engineer at the University of Oxford, was editor-in-chief of Neuroimage and now holds the same role at the new journal, Imaging Neuroscience, which launched in July 2023. The resignations are a symptom of the tension between the competing priorities of publishers, who demand constant growth, and researchers, who value quality and depth. Many editors are taking matters into their own hands by founding new journals that are open, not-for-profit, and have high academic standards. The resignation is not so much the point. The point is creating an alternative top-quality channel of scholarly communication, says Klaus Arbels, a linguistics researcher at University College London and one of the editors who resigned from Syntax. While it is unclear whether mass resignations will become even more frequent, they are certainly getting a lot of attention. Michael Clark, a publishing consultant at Clark & Esposito in Washington, D.C., says that many mass resignations are in response to changes to business models in the publishing industry. If an academic community wishes to control the business decisions of a journal, the best way to do that is to own the journal, Clark says. Sources: *Journal editors are resigning en masse. What do these group exits achieve? Nature.com. Editors quit top neuroscience journal to protest against open access charges. Nature.com. Open access row prompts editorial board of Elsevier journal to resign. Nature.com. Retraction Watch. Retractionwatch.com. URL. Less than nature greater than.